Thanks everybody for showing up. My name is John. I'm a postdoc in Jim Collins' lab at MIT and the Broad Institute. And I'm going to quickly summarize a paper that we just published 2 months ago now, wherein we leveraged some ML approaches to help us discover new antibiotic molecules. This is a slide I like to show that kind of summarizes our history of antibiotics. We've had antibacterial molecules for the better part of a century, and the discovery process throughout this 100 years has kind of gone through a number of different eras. Perhaps the most productive of these eras lasted from the 1940s to the mid-1960s. This is defined as the golden era of antibiotic discovery. And during this time, we witnessed, a lot of productivity in discovering novel structural and functional classes of antibiotics through screening secondary metabolites that were produced by soil-dwelling microbes. So here we would isolate microorganisms from soil and then screen extracts of these microbes for secondary metabolites that exhibited antibacterial activity against a pathogen of interest. However, you can see by looking at this graph that by the mid-1960s, our ability to discover novel antibiotics through this approach was diminished. We kept discovering the same antibiotics over and over again. So in more recent decades, we've turned to high-throughput screening of synthetic chemical libraries for new antibiotics. So what that looks like is, again, you have a pathogen of interest, and you curate a chemical library that might be, hundreds of thousands to maybe a couple million molecules, and then you screen these molecules against your pathogen of interest to see which ones have any microbial activity. Now, this process is slow. It can take months or years to screen a large chemical library. As you can imagine, it's also very expensive because you have to physically curate, the hundreds of thousands to millions of molecules you want to screen. And a lot of the times, the chemicals that you can acquire for high-throughput screening don't explore a vast chemical space. So because of these limitations, unfortunately, we have not discovered any new clinical antibiotics using high-throughput screening approaches. So kind of with this history in mind, our team in Jim Collins' lab, as well as the team who developed ChemProp in Regina Barzilai's lab, both at MIT, we wondered if we could leverage ML approaches to help us discover new structural and functional classes of antibiotics. So instead of screening, being upper bound by maybe a couple million molecules in our search for new drugs, could we train an ML model and then have the capability of exploring tens of millions, hundreds of millions or more molecules that may have antibacterial activity? So that's what we did. The first step was to gather training data with which we could train a model. So we actually curated a library of about 2,500 molecules. These were FDA-approved drugs, as well as a purified natural product library. And then all we did is we took these 2,500 molecules and then assayed to see which of these did or did not inhibit the growth of E. coli in the lab. And that's what we're seeing here. All the molecules in blue are molecules that we called as non-growth inhibitory, and then all the molecules in red are those that did inhibit the growth of E. coli in the lab. Everything that was not growth inhibitory, we labeled a 0. Everything that was growth inhibitory, we labeled a 1. And this is what we used to train a ChemProp model, right? So we had the graph structure of every molecule tied to an activity score, either 0 or 1. We trained the structure property predictor. And therefore, when we showed the model a molecule that it had never seen before, it would predict whether or not it was antimicrobial. A prediction score closer to 0 would mean that it was highly unlikely to be antibacterial. A prediction score closer to 1 would mean that it was more likely to be antibacterial. So once we had gathered the training data and trained the ChemProp model, we first used our model to run predictions on the Drug Repurposing Hub at the Broad Institute. So the Drug Repurposing Hub is a collection of about 6,000 molecules, and these are compounds that range from preclinical investigations all the way up to compounds that are on the market for treating a variety of diseases. So here at the bottom of the slide, we can see all the molecules that were unique to the Drug Repurposing Hub and their corresponding prediction scores ranging from 1 to 0. So what we did to see how well our model was able to make predictions is we took the 99 molecules that had the highest prediction scores and then curated them and then tested them in the lab. We wanted to see how many of these top 99 predictions actually displayed growth inhibitory activity against E. coli when we tested them in the lab. So of these top 99 molecules, we observed that 51 displayed antibiotic activity. And then we asked the next question. We said, okay, so we have 51 molecules that are growth inhibitory. Of these 51 molecules, are there any that don't look like existing antibiotics, right? Because we were looking for molecules that were structurally and functionally unique relative to anything that we have. So the molecule out of these 51 that fit that criteria was Halicin, which is shown on the right. So given that we observed this molecule that was strongly predicted to be antibacterial, it validated in the lab, and it didn't look like any conventional antibiotics, we wanted to explore its biological properties a little bit further. So the first set of experiments that we did were, again, just using lab strain E. coli. On the left, this is just dose response of growth inhibition of E. coli with Halicin. And we observed that Halicin can inhibit the growth of E. coli at about 2 micrograms per milliliter concentration. And we observed, additionally, that this growth inhibition was due to bactericidal mechanisms. So we see on the right that with increasing concentrations and increasing time, that Halicin is able to kill E. coli in culture rather than just inhibiting its growth. So given that we observed that Halicin displayed bactericidal activity against E. coli and that it was structurally distinct relative to our current collection of clinically used antibiotics, we wanted to see whether Halicin would retain activity against antibiotic-resistant pathogens. So what we did is we collected 36 isolate panels of carbapenem-resistant Enterobacteriaceae, Acinetobacter baumannii, and Pseudomonas aeruginosa. These are 3 clinically problematic gram-negative species that are often multi-drug resistant. And what we observed was that Halicin retained activity against both carbapenem-resistant Enterobacteriaceae as well as multi-drug resistant Acinetobacter baumannii. Unfortunately, we observed that Halicin did not display activity against Pseudomonas aeruginosa, and this is likely a result that can be attributed to the fact that the permeability barrier to get molecules into Pseudomonas aeruginosa is great. Furthermore, we tested Halicin against Mycobacterium tuberculosis. So this is an extraordinarily slow-growing bacterium that often requires antibiotic administration for many months in order to clear an infection. And we observed that Halicin was able to eradicate cultures of MTB in just about 24 hours. This was a very striking result, and we are currently studying the mechanism underlying why Halicin was able to eradicate TB so quickly. So given that we observed excellent Halicin efficacy across a broad range of clinically problematic pathogens, we next ventured to understand the mechanism. So what we initially tried to do was to evolve spontaneous resistant strains of E. coli. Because the idea would be we would evolve resistance, sequence the resistant strain, and then identify the mutation that conferred resistance to help point us in a direction of what the mechanism of action underlying Halicin's efficacy is. However, looking on the left, we can see that over a 30-day evolution experiment in liquid culture, we were unable to evolve resistance to Halicin, where in stark contrast, we were able to readily evolve high-level resistance to the fluoroquinolone antibiotics ciprofloxacin. On the right, we can see an experiment that was ran in parallel, where we attempted to evolve Halicin resistance on solid media over the course of 7 days. And again, we were unable to isolate spontaneous resistant strains of E. coli. So what we turned to was RNA sequencing to elucidate mechanism. Here we took wild-type E. coli, treated it with a range of concentrations of Halicin over various durations, and then did whole cell transcriptomics. And the most striking feature that we observed from the resultant data was that cells immediately downregulated genes involved in locomotion, flagellar biosynthesis, and membrane protein complex biosynthesis. A quick literature search thereafter revealed that genetic downregulation of genes involved in locomotion and flagellar biosynthesis are often the result of dissipation of the proton motive force. So to verify this hypothesis, we employed the dye DISC-35, which is a potentiometric fluorophore that allows you to measure proton motive force across membranes. And we observed upon treatment with increasing concentrations of Halicin, which are shown here in blue, that Halicin administration to cells immediately caused a decrease in DISC-35 fluorescence, which is indicative of Halicin dissipating specifically the pH component of the proton motive force. So that's the proton gradient across the membrane. So once we had this, reasonable mechanism of action, we next asked, does Halicin display any reasonable antibacterial efficacy in vivo? So to test this, we set up two infections in mice. The first one was an Acinetobacter baumannii skin infection. So baumannii is a problem for soldiers, wounded soldiers, particularly in the Middle East. And Acinetobacter, this pathogen is particularly worrisome because of its propensity to acquire and maintain antibiotic resistance. So what we did is we took a multi-drug resistant Acinetobacter strain, infected a wound on the dorsal surface, so on the back of mice, administered Halicin topically for 24 hours, and we observed that Halicin was able to eradicate the baumannii infection over the course of 24-25 hours. The second model that we tested Halicin in was a C. difficile gut infection model. So C. diff is an opportunistic pathogen that causes diarrheal diseases, and you'll often see patients with C. difficile infections after they have taken systemic antibiotics. So systemic administration of antibiotics will decimate the native gut microbiota, allowing C. difficile infections to bloom. And this is another urgent pathogen that we need to develop therapies for. And indeed, in the U.S. alone, there were more than 223,000 cases and 13,000 attributed deaths in 2017. So we can see on the right-hand side of the slide, we established C. difficile infections in mice and then treated orally with Halicin once a day over the course of 6 days. And we could see that 3 to 4 days after administration of Halicin, we were able to eradicate the C. difficile infections in the mouse gut. So given that we had these exciting results with Halicin, we next asked, instead of looking for new antibiotics in relatively small libraries, can we now apply our model to search vast chemical spaces that we would not be able to empirically screen, right? So for this, we turn to the Zinc-15 database. So the Zinc-15 database is a virtual collection of about 1.5 billion molecules. Instead of running predictions on the entire set of 1.5 billion molecules, we curated about 107 million molecules with antibiotic-like physical chemical properties on which to run predictions. So after curating these 107 million molecules, we ran predictions. And again, we prioritized molecules that were both strongly predicted to be antibacterial, as well as those that were structurally distinct from known antibiotics. We were able to curate 23 of such molecules, shown on the bottom left. And excitingly, 8 of these 23 empirically assayed molecules validated as antibacterial. Of these 8 molecules that empirically validated, 2 of them, shown on the right, displayed broad spectrum activity across a wide range of clinically burdensome bacterial pathogens. And these 2 molecules are currently undergoing investigation both for mechanism and potential in vivo efficacy. So with that, I would like to thank you all for listening, funders, a large team of amazing collaborators. And I believe now we are open for questions.
Yes, John, thank you so much. So we are open for questions. As a reminder, you can chat us your questions in the chat box on the right side of your screen at the bottom. Send your questions to all panelists. Type in your question, hit enter, and that'll send it our way. In the meantime, I've already got several that we collected during the talk. So I guess we'll start with you, John. What are the features feeding into the learning network? Are they chemical bonds, atoms, or just the graph of the formula?
Yeah, so how we did it is we curated smile strings for every single molecule that we were interested in. And then during training, those smile strings are converted into molecular graphs, so the actual structure of the molecule. So what ChemProp is learning is the bioactivity of the molecule in relation to its 2-dimensional or 3-dimensional structure. And in contrast to previous methods, like older methods, to predict biological functions of molecules, our approach was never taught what chemical features to look out for. There was no manual curation of, this is a nitro group, this is, some aromatic heterocycle. The ChemProp itself learns what molecular features of each molecule are important for its corresponding bioactivity or lack thereof.
Great. So we're going to hop around a little bit because I've got questions for most everybody. So for Adam Marco, in Igneous, can you write to any cloud tier directly, or do you have to stage data?
Thanks. We're one of the few products that can write to any cloud tier directly, so we don't have to do an S3 initial transfer and then move it from S3 or tier it off using S3 commands to get it to a cheaper tier. So we can go directly to Glacier Deep Archive, Azure Archive, and the cold tiers in Google.
Great, thank you. All right, the next one's for Fernanda. NVIDIA has a huge footprint in the GPU analysis space. Are there open source tools you recommend that can seamlessly use mixed environments with NVIDIA and AMD GPUs?
So I want to remark that I'm no longer at NVIDIA, so I cannot speak for NVIDIA, but there are many tools that allow you to use both AMD GPUs in terms of programming language, for example, OpenCL. The question is whether or not those tools are available cross-platform. And that still is building up. AMD is just currently moving to the DOE compute space, and that is going to create a nice amount of tools and available programming languages that can probably communicate with both. But that still remains to be seen whether it'll be mature enough within maybe the next year.
Great, thank you. Here's another one for Adam Marco. How do you manage permissions across all the different platforms?
Okay, so I'll answer this the best I can from a technical standpoint. We have our own lightweight NFS and SMB clients. We don't use something like NDMP. We don't run an agent on your servers or your storage system. So from that, we actually just pull the permissions as they are and store that in our scalable file system metadata index. So in that way, we know the permissions when we're storing it to your backup destination, and then, or when we're sending it to your backup destination, and when we restore it, all those permissions are preserved.
Great, thank you. Giving Jeff another second if you want to jump in.
No, I think Adam stated it correctly, is that we have full visibility of the permissions, and we find that folks have differing policies around whether they want to use file system permissions or if they have kind of lab level permissions that they like to use based on a directory structure or whatnot. So we're flexible in how you want to use the permissions to have visibility to folks or not.
Great, thank you. Another sort of interoperability question for Igneous. Can you work with any NAS platform?
Yeah, this is Adam. Marco, I can answer that. We know that pretty much every organization has some mix out there, whether it's an Isilon or GPFS or home-built JBOD. As long as you can export NFS or SMB, we can scan that and back up and archive it as a source appliance.
Great. Okay, thank you. This had come up, I think Adam Kraut mentioned briefly the ontology issues, but could you maybe explain further, Adam Marco, how Igneous deals with ontology issues?
Yeah, I'll answer that from a product and infrastructure standpoint, and then I'm actually going to also ask BioTeam to weigh in on that from more of a scientific standpoint. So we scan and analyze your file system using file system metadata currently. And there's a number of things you can do. I did show some of those live views, and there's a large number of attributes we can present to the user, and then they can share and visualize. Now, what the person asking the ontology question may be asking is about user-defined metadata or custom metadata. As a former scientist, I absolutely understand the need to be able to apply custom metadata for research and for collaboration. And as data sizes grow, it makes sense. There's a lot of organizations trying to do that. I feel like very few have got it really right. We're focusing on how to do that intelligently and not compromise performance or the user experience. We're already working on extracting metadata from common life science files like FASTQ or Illumina sample sheets or some cryo-EM files as well. So you'll see that showing up in our application. From a more scientific research and planning and going forward standpoint, I'm going to see if Adam Kraut or Fernanda have any more input in that area.
Yeah, I mean, I'd say in terms of ontologies, there's not a one-size-fits-all solution. Obviously some organizations on the low end will benefit from, just simple metadata policies and standards. So, setting a minimal amount of tags you want to see in whatever metadata system you're using, right? Whether it's tags on an S3 object or something like iRods or some other storage, right? But the other thing to think about is the domain-specific consortia that come up. So things like the Global Alliance, right? Some of their API specifications of things are obvious things to adopt if you're kind of working in that same space. So I'd say looking to communities where you can align with standards, but then also doing the simplest thing first for your team is also pretty pragmatic.
Great, thank you. We do have a sort of domain-specific question. Next, is there a pilot established in a specific cloud, AWS for example, to facilitate COVID-19 with NGS data, antibodies, drug information to stimulate collaboration and sharing scientific insights? I keep hearing a lot about different coronavirus and COVID-19 collaboration. So are there any that y'all can speak to specifically, perhaps, that are maybe cloud-based?
So I can take a stab at that. There is for computation, there is a HPC consortium on the United States side that allows folks to request for compute time. Now, what they're asking here particularly is data. And data, the concentration of those types of data into a single location does not exist. And this is where folks that are working on multimodal data are going to have to build the tools probably themselves as they understand how to collate these types of disparate data together. But they reside with the source that created them, or they are in a place where they're siloed and it's difficult to combine.
Thank you, Fernanda. Anybody else want to jump in on that?
Yeah, another one that I can plug is there is a COVID-19 specific data commons that's being built by the University of Chicago. It's based on the Gen 3 platform, so it's all cloud-based, and that's pandemicresponsecommons.org. And so that's attempting to bring together a lot of genomics data as well as some of the data related to the pandemic that's coming in. So check that out. That's open and online now. I can share the link in chat.
Yeah, that'd be great. Thank you. I know MBI has one that they announced last week as well. So yeah, there's a lot of folks coming together trying to share data around this particular situation, which is great. But I think we're going to wrap it up. We've got lots of good information to go through here. I want to thank Adam, Fernanda, and John so much, and Jeff for piping in as well. Appreciate all of your input. For those of you listening, again, you will get an email link to this to re-listen, check some things, maybe check on the slides if you'd like, and to share this with colleagues. So I hope that you will do that. I believe everybody also offered their own email addresses. If you want to follow up with further questions, you can do that directly. And that's it. Thanks, everyone, so much for joining us, and thank you again to our panelists and our sponsors.